Hi everyone and welcome to my channel Be Yourself. This is Dr. Rajni Sharma and today I'm going to discuss the answer key as well as I will explain the different questions of part C of CSIR JRF Net Life Sciences, which was held on 18 December 2016. I know that you all are waiting for the part C section because it is assumed that it is the toughest part of the life science. But it is not so. It is totally based on the concept and the answer is there itself in the question paper. How? Let's see. The first question of the part C is a researcher was investigating the substrate specificity of two different enzymes X and Y on the same substrate. Both the enzymes were subjected to treatment with either the heat or an inhibitor which inhibits the enzyme activity. Following are the results obtained where A is the inhibitor treatment, B is the heat treatment and C is the control. Now come to the graph. When you will see this is the control, A is the inhibitor treatment and B is the heat treatment. In the both cases, if you will analyze that it's being a control, then if it should not be affect or we can say if the inhibitor will not affect the activity, then it should have the same curve. But in the both cases, if you will look in both the graph or with the protein X and Y, the graph is different from the control graph C. So both the enzymes are based on the are specific for the substrate, because if it get inhibited it is showing different graph from the expected. So the answer will be both X and Y are specific for the substrate. The following are the four statements of peptide protein conformation. First is the glycine has the largest area of conformationally allowed space in Ramachandran plot phi and psi. It's true because if you will remember, I have explained during the explanation in the part B of the Ramachandran plot that glycine, because of the absence of any stirring hindrance from the R group, it has very large area in the Ramachandran plot. So this statement is true. Next is a 20 residues peptides that is acylated at the N terminus and amidated at the C terminus has psi minus 60 degree and phi minus 30 degree for all the residues. It is not. It can be concluded that conformation of the peptide is helix turn strand. Actually, helix structure is not lies in this range, so this statement is false. The allowed values of phi and psi for amino acids in a protein are not valid for the short peptides. It doesn't matter whatever the size of the peptide, it will be valid for the all types of peptide. Third is a peptide acetylated at the A1, A2, A3, A4 and its C terminal get amidated, and this A2, A4 is the amino acid and adopt a well-defined beta terms. It's 2, and because here it is written that dihedral angles of A2 and A3 determines the type of beta turn, because it actually the if glycine will be there and proline will be there, then there will be different conformation in the case of the beta turn. If you beta actually proline, which has a very large bulky group, it will leads to the hydrogen bonding different way, and the glycine will lead to the hydrogen bonding different way. So the answer will be D. Both is true. A and D is true. So the answer will be number 3. Next question is, from the following statements hydrogen, deuterium, tritium differs in number of protons. No, it differs in the number of protons. So in the second statement it written that it differs in the number of neutrons. So this statement is true, and both deuterium and tritium and are radioactive and decay to hydrogen and deuterium respectively. It doesn't, so rather tritium will be decay to the helium. So the statement D is true, and likewise carbon 14 will be decay to nitrogen 14. So the answer B, D, E is true. So the option will be number 2. In the biosynthesis of purine, I'm not going to discuss this question because it is completely based on the chart or the biosynthetic pathway of the purine. From there, you will come to know that N1 is from the aspartic, N2 is from the glutamine side chain, and whereas another 7 and 7 is from glycine, and 9 is from the alpha C alpha of the glutamate, where C4 and C5 is also from the glycine. So the option will be number 3. Next question. Yes. The researcher investigated a set of conditions for the protein with the isolectic point of 6.5 and also binds to calcium. This protein was subjected to 4 independent treatments with a pH 6.4, 10% of glycerol, 10 millimolar of calcium chloride, and 40% of the ammonium sulfate. This is followed by the centrifugation and estimation of the protein in the supernatant. The results are depicted in the graph below. The condition where the protein will be high in the supernatant, it means that when you are treating with any of the above conditions, and if the protein get completely precipitated in that condition, then it will be settled down at the bottom of the centrifugation tube after centrifugation. But if it is not get fully precipitated, it will be present in the supernatant. So before that, we must have to find out which condition will completely precipitate the protein. As here isoelectric point is given 6.5 and the pH is here 6.4. So this is very much close to the isoelectric point where the pH is 0. So this will completely precipitate the protein. And second is the 40% ammonium sulfate, which will completely precipitate the protein by salting or process. But in the case of calcium chloride, which is also required for the precipitation of protein, but being its very small quantity is used here, which is 10 millimolar, it will not completely precipitate the protein, and the glycerol too, it will not completely precipitate the protein. So where the supernatant will be high, it will be in the glycerol and calcium chloride. So either these two bar graph may have represented the calcium chloride and glycerol, and either of the two, B and D, have represented the pH 6.4 and 40% of ammonium sulfate. So if you will look all this, and you will come to know the answer will be number 4. Next question is, from the following statement, for a reaction to occur simultaneously, the free energy change must be negative. It's true, because for the spontaneous reaction, del G is minus, and for equilibrium it is 0, and for a non-spontaneous reaction, del G is positive, or the Gibbs free energy is positive. The interaction between two nitrogen molecules in the gaseous state is predominantly electrostatic. It is not so. We can't say that if two molecules in gaseous state, it will be electrostatic. By knowing the bond energy, it is possible to deduce whether the bond is covalent bond or hydrogen bond. Yes, with the help of the bond energy or the Gibbs free energy, we can deduce the nature of the bond. Last is hydrophobic interaction are not important in the folded global protein. It's not so. Hydrophobic interaction is very much important for the folding of the global protein. So what we have to do here, pick up the combination of all wrong statements. So the wrong statement is number B and D, and the option will be number 4. Next question. Addition of antibiotic cephalexin to growing E. coli cell leads to filamentation of the cells followed by lysis. So what will be the inhibitor points of the cephalexin? Without any doubt, it will be peptidoglycan synthesis, because if you will see here, there is the lysis of the cell, and this is only possible when the cell wall get lysed, and so it should affect the peptidoglycan synthesis to lyse the cell. So without any doubt, the answer will be peptidoglycan synthesis. Next question is, fluorescent tag protein was used to study protein secretion in yeast. Fluorescence was observed in Golgi body, in secretory vesicles, and rough endoplasmic reticulum. What happens Protein is synthesized over the endoplasmic reticulum, and from here, according to the signaling molecules attached to the protein, it gets transported to the required places. So, what happened? First of all, protein from the endoplasmic reticulum get transported to the ergic or the endoplasmic reticulum uh, Golgi body intermediate complex, which is ergic, and from there it is transported to the outer side of the trans side of the Golgi body, and from there to the place where it is required. And during this process, the protein undergo modification, which can be post-translation, which is known as post-translation modification. And after recognizing the proper signal molecules, it gets transported to the place where it is required. So the pathway should be first from the rough endoplasmic reticulum to the Golgi body, then to the secretory vesicle. So the option should be C, A, and B. So the answer will be 3. Next question. In order to ensure that only fully processed mature messenger RNA is allowed to be exported to cytosol, pre-messenger RNAs associated with snRNPs are retained in the nucleus. To demonstrate this, an experiment was performed where a gene coding a pre-mRNA with a single intron was mutated either at the 5' or 3' splice sites or both the splice sites. To remove the introns, suppose this is the RNA and this portion is the intron, suppose this is the 5' end and this is the 3' end of the intron. When you will cut at the one end, then even in that position, intron is in with the messenger RNA. When you will cut both the side, then it is possible that this intron will be removed and the exon part will be then ligated. So to export the messenger RNA or the matured messenger RNA, we should cut the both side of the intron. Now move to the options. First is pre-mRNA having mutated at both the splice site will be retained in the nucleus because of the presence of bond. It is not so. If both sides are cut, it means we have now only the exon parts. After its editing, it will be transported to the cytoplasm, not it will return to the nucleus. So it will be wrong. Pre-mRNA having mutation at both the splice sites will be exported to the cytosol. It's obviously if both the sites are cut or removed, it means the only exon parts will be there. So this will be transported from the nucleus to the cytoplasm. Next is pre-messenger RNA mutated at either 5' or 3' splice site will be retained in the nucleus. Yes, because if only one site will be cut down, then introns is still present there. It will not be removed. So it will be retained in the nucleus for its further processing to remove the introns part completely. So the answer will be B and C, and option it is option 1. Question number 80. Yes, it's a very simple question, but by looking at this figure 600, 100,000, you may have confused or may have panicked of what it is asking, but it is not. So it is very simple question. So have a look. In an experiment, red blood cells were subjected to lysis and by unbroken cells were removed by centrifugation at 600 gram. The supernatant was taken and centrifuged at 100,000 gram. The pellet was extracted with the 5 molar NaCl and again centrifuged to 100,000 gram. Which of the protein would be present in the supernatant? The protein which will be inside the cell will be present in the supernatant, and the protein which will be in the along with the plasma membrane will be at the will be centrifuged and it deposit as a cell debris. So if you will look the band-free protein glycophorin and G protein couple receptor, all these are the membrane bound proteins. Only the spectrin is the peripheral protein. So when we will treat with any of this chemical, this will be detached from the membrane, and on centrifugation, these 3 protein will be settled along with the plasma membrane at the bottom of the centrifugation tube, while the spectrum will remain in the supernatant. So the answer will be number 4. Next question is, in order to study the intracellular trafficking of protein A, it was tagged with GFP. And what is GFP? It is a green fluorescence protein, and the fluorescence microscopy shown that AGFP co-localized with LAMP1 in the presence of bapillomycin A, an inhibitor of hydrogen ion ATPase. AGFP does not localized with the LAMP1. Instead, it co-localized with the LC3 puncta. Which one of the following statement is true? This is also a very simple question, but you might have confused because you were not knowing the full form of this LAMP and LC3. What the function of all this? In the question itself, it is mentioned that bapillomycin is the inhibitor of hydrogen ion ATPase. Look, I have told that you might got panic with the part C, but the part C is the easiest part if you have once you have the concept of all these things. Now, what is the LAMP1? LAMP1 is lysosomal associated membrane protein. Now, it means this will be this trafficking molecules is trafficking or traveling toward the lysosomes. Now leave it. Now it is not localized with the LAMP1, so it means it is not going to the lysosomes. Now, but it is co-localized with the LC3, and this is a marker protein that is involved in the formation of autophagosomes and autolysosomes. It means it is again traveling towards the lysosomes. So, and it now is this time it will lead to the formation of autophagy or the autophagosomes. So, without any doubt, the answer will be autophagy is required for trafficking of AGFP to lysosomes. Question number 82. A is the inhibitor of chloroplast function. The production of oxygen and synthesis of ATP are measured in illustrated chloroplast before and after the addition of A as shown below. Now, if you will see these two graphs, then on addition of A, the oxygen production doesn't get hampered. But in the case of graph 2, addition of A is decreasing the production of synthesis. Now, if you will look at the chemoosmotic theory, it is the intermembrane space of the mitochondria, and this is the proton pump, and this is the ATP synthase. What happened with the transportation of NAD positive towards the intermembrane space where the hydrogen concentration is very high? This get to maintain the potential difference. This hydrogen, some of the hydrogen get imported inside the membrane, and during this process ADP is get converted into ATP. So if ATP synthesis is decreasing, it means it is inhibiting or reducing the reduction of NADP positive. But proton gradient pump, so it is inhibiting the proton gradient pump but not the NADP positive, which leads to the production of oxygen during the ETC cycle, which is the electron transport chain cycle. Okay, so the answer will be A. A inhibits proton gradient but not the reduction of NADP positive. Next question. Sorry. During cell cycle progression from G1 to S, cyclin D-CDK4 phosphorylates RB, which is the retinoblastoma protein, and reduces its affinity for the E2F, which is elongation factor. E2F dissociates from RB and activates S phase gene expression. Look, the question itself has answered all the things. Now you have just to analyze which option is correct. So I think if you will know the trick behind this, part C will be the easiest part for you. Overexpression of the protein A arrests G1 phase progression. Which of the following statement is true? Look, overexpression of which protein will arrest the G1 phase progression? It means it can't cross the G1 to S. What happens during the G1 phase? When RB is free or it is not phosphorylated, it will bind to the elongation factor and it will prevent the transcription. But when it has once crossed from G1 restriction point and entered to the S phase, then it will be phosphorylated with the help of CDK4/6 cyclin D, which is the checkpoint from the G1 to S phase. Sorry, G1 start point. Okay, I have discussed this in one of my video during the cell cycle. It is there in my channel. You can get that from my channel. Now after it gets phosphorylated, it will unable to bind with the E2F, and now E2F will able to transcribe and can enter the S phase. So if there will be restriction of the G1 phase, it means it will inhibit the CDK4 activity, which is phosphorylating the RB. So once it get inhibited, it will not able to phosphorylate RB, and it will get inhibited at the G1 phase or arrested at the G1 phase. So the answer will be A, inhibit CDK4 activity. Next question is, cell in S phase of the cell cycle were fused to cells in the following stages of the cell cycle: G1 phase, G2 and M phase. These cells, which were then grown in the medium containing titrated thymidine. Maximum amount of freshly labeled DNA is likely to be obtained in S-shaped cells fused with. Maximum number of labeling will occur when when it will fused with the G1 phase cycle, because while it has crossed from G1 to S phase, the synthesis or the replication of DNA starts, and during the replication it will require the raw material or the thymidine, and during replication this will be fused with the DNA. So it will fuse with the G1 phase, it will have the maximum number of thymidine, titrated thymidine. But when it will get fused with the G2 phase, it has already most of the DNA get replicated, so it will not have that to contain the thymidine, or it will not absorb the thymidine from the medium. So the answer will be G1 phase cells. Next question is telomerase. A protein RNA complex has a special reverse transcriptase activity that completes replication of telomerase during DNA synthesis. Although it has many properties similar to DNA polymerase, some of them are also different. Which of the following properties of telomerase is different from that of the DNA polymerase? If you have seen my explanation of the part B, on that time I have discussed how polymerase get attached to the leading strand, not the lagging strand. It get attached here and replicated some of elongated to some extent this leading strand, and after that primer get attached and the unreplicated strand get replicated, and it has resembled with the original one. So the answer will be telomerase doesn't carry out the lagging strand synthesis, rather it only carry out the leading strand synthesis, whereas DNA polymerase replicate both leading and lagging strand. So the answer will be telomerase does not carry out lagging strand synthesis. Next question. In eukaryotes, a specific cyclin-dependent kinase activity is required for the activation of loaded helicases to initiate replication. On the contrary, this CDK activity inhibits the loading of helicases onto the origin of replication. Considering the fact that during each cycle there is only one opportunity for the helicases to be loaded onto the origins and only one opportunity for these loaded helicases to be activated, which one of the following graphs best depicts the CDK activity in G1 and S phase of the cell cycle? When it is activating the replication, it means it will be synthesized during the S phase, not the G phase. Okay, so the answer will be 1, where its concentration is very low at the G1 phase and get picked up during the S phase. Next question. Polysome profiling of cells treated with 3 hypothetical translation inhibitors is shown in the plots below. These 3 inhibitors are CHP, which is the leaky inhibitor of the translation, LTM, which arrests the ribosome at the initiation codon, and PTM, which inhibits ribosome scanning. what happened during the initiation process? First of all, suppose this is the messenger RNA. First of all, 30S ribosomes get attached to the initiation. It will first scan the upstream region of the messenger RNA, and once it gets the initiation site, which is the AUG sequence, it will get attached or get fixed at this point. After that, tRNA along with its anticodon will be attached to this AUG sequence, and after that the initiation complex get attached with the 50S ribosomal RNA, and now it completely form the initiation complex. Now move to the question. Here, leaky inhibitor of the translation. It means that uh, it uh, it has jumped out from the initiation site. So once it hasn't found the initiation site, and being this is a polysome, there should be multiple polysomes in the regions because it is not recognizing the initiation site. So there will be multiple ribosomes along with the 30S and 40S ribosomes, and the once the initiation complex has formed. So there will be the four concentration. One will 40S, 60, 80, and polysomes. So one will be the graph for the A. Move to arrest ribosome at the initiation complex codon. It means if it is arresting at the initiation complex, it means it might be possible that the 50S ribosomal subunit has not been attached at the initiation site. It get attached once after the tRNA has attached along with the anticodon with the AUG sequence. So it will have the free 50S and 40S subunits. 60S. I have written 50S. Sorry, it will be 60S. 60S subunit. Okay. So there will be no complex formation of initiation complex, or the in fact ATS will not form during this process. So the graph will be number C. Number 2 will have the number C graph, and lastly arrest ribosome at the initiation codon inhibits ribosome scanning. When it is preventing to scan the upper stream region, then it will not be able to translate it. So in that case, might be possible that whole ribosomes can be present, and the subunits will be present. So BV will be for the number 3. So the option will be number 4. Move to the next. In mammals, CG-rich sequences are usually methylated at C, which is a way for marking genes for silencing. Although promoters of housekeeping genes are often associated with the CpG island, as they are expressed in the mammal. Which of the following statement best explains this? Why this housekeeping genes expresses in mammal? Because the promoter regions usually have the where the active genes is present, it doesn't get methylated. So the answer will be, unlike within the coding region of a gene, CG rich sequence present in the promoter of the active genes are usually not methylated. Now this question, without any explanation, we have to match out the function of this column A with the column B. And integrin is the we know the transmembrane protein, or it is extracellular matrix receptors. Cadherin is the number C, which mediates. Cadherin is calcium mediated a strong hemophilic cell to cell adhesion. It helps in the adhesion of the with this nearby molecules. And immunoglobulin Ig superfamily is named because of this. It's as it contains extracellular immunoglobulin like domain, so it is named because of this immunoglobulin like domain. It is named as immunoglobulin superfamily. And selectin, and if you have remember, it is the I have explained in this question, this part also, that spectrin is present in the RBCs, and it is the peripheral protein, and it acts as the cell adhesion molecules in the bloodstream. So the answer will be number 4. Last question of this video is, The student treated cancer cells with the anti-cancer drugs and performed Western blot analysis. Which one of the following blot is the best representation of the treated, control and tested T sample? This band shows the control and this band shows the treatment. If it is cancerous cell, then in that case the growth factor should be high, which is the cyclin D1, which regulates the cell cycle, and another is the HEDGEHOG. It is basically responsible for the digit formation or the limb in the limbs, or the finger formation in the limbs, finger or toes formation in the limbs in the case of humans. So in the case of mammals, so in the, this graph shows, this band shows the cyclin D1, and this sonic HED factor is high in the human, in the patient. So when this will get anyway, if this need the treatment, on treating it, there should be high level of P53, which is involved in apoptosis, and cleaved PRP factor, which is also involved in the for the stop, or it halt the process of the cell cycle. So the option will be number 3. So this was in this video. I have discussed first 20 questions of the part C, and if I will discuss some more, you will get bored out and might feel sleepy. In my upcoming videos, I will discuss all of the rest questions of the part C. I will not delay much in explaining all these questions. I have only selected only the point wise. I am not discussing much about the part C, but some have requested to me to discuss the past few years question paper of part C. I'm also preparing for that, but I think it will be much better if I will discuss some of the expected questions which are usually asked in the CSIR net or other competitive examinations. So don't forget to subscribe to get updated with that. Till then, goodbye, and don't forget to share my channel so that all get benefited. And goodbye, be yourself.